Hi friends, welcome back to my channel. I will introduce you today with one form of microorganisms that is called bacteria. So here is a bacterial cell. It contains cytoplasm and all the organelles and nuclear material of bacteria is present free in the cytoplasm. It is not enclosed. The nuclear material is not enclosed, and there are flagella and pili which help for the movement of the bacterial cell from one place to the other place. Now, bacterial cells are prokaryotes, which means their nucleus is open, it is not enclosed, and it is openly present in the cytoplasm. And these are unicellular organisms. So, these bacteria lack well defined nuclei and membrane bound organelles, and with chromosomes composed of single closed DNA circle. They come in many shapes and sizes, from minute spheres to cylinders and spiral threads, to flagellated rods and filamentous chains. They are also found practically everywhere on Earth and live in some of the most unusual and seemingly inhospitable places. So, here are some shapes of bacteria, various shapes like cocci, bacilli, and other shapes are also there. So in cocci also they come either in duplicate two cells together, that is diplococci. Then they can form a chain, streptococci, or they come in groups called staphylococci, and they can also be found in tetrad or in six cells group called sarcina. So bacilli also they can be unicellular or they form a streptobacilli you can say. Then, whether bacteria are good or bad, bacteria are essential to human life and life on planet Earth. They are the most primitive organisms. The life probably started from viruses and bacteria only. So, although they are notorious for their role in causing human diseases like tooth decay to the plague and various other diseases, they are also beneficial species that are essential to good health. So, here are some useful bacteria. Some bacteria manufacture vitamin K, which is essential for blood clotting factor, and it is present in our intestine. The bacteria are present in our intestine, which manufacture this vitamin K. Bacteria also provide yogurt its tangy flavor and bread its sour taste. They also make possible ruminant animals to digest plant cellulose, and for some plants, they convert nitrogen to more usable form, like rhizobium bacteria. Some bacteria produce antibiotics and vitamins and enzymes, which are used in several medicinal preparations. So, in fact, bacteria are good, uh, but there are some bad bacteria also, like harmful bacteria. Here are some diseases which are listed, but the list is a small list. There are many other organisms, bacteria, which cause various diseases. So, cholera, typhoid, pneumonia, tooth decay, diarrhea, skin infections, intestinal ulcers, UTI infections, and several other infections are caused by bacteria. So, where you will find them, these organisms, bacteria, they are ubiquitously present in all forms of elements, air, water, soil. 1 gram of soil contains uncountable bacteria, and millions of bacteria reside on the surface of human body and also in alimentary canal. Most of them are symbiotic, beneficial association between man and bacteria. So symbiosis is happening between bacteria and human beings. Now coming to the last part, that is gram staining. So this is the technique which is used to classify bacteria into gram positive and gram negative organisms. So in this technique, what is done is on a glass slide, you have to make a smear of bacteria. That is, you have to spread a droplet of suspension of bacteria and dry it, and then add crystal violet indicator solution and allow it to stain for some time. Then you need to add iodine. Iodine is used as a mordant, so it will form crystal violet iodine complex. And after that, you have to add alcohol, which acts as a decolorizing agent. So basically what happens is gram staining. In this technique, gram positive bacteria are stained violet in color because they contain peptidoglycan in their cell wall. So they are able to retain crystal violet. Whereas when you add alcohol, gram negative bacteria release this crystal violet iodine complex and they become decolorized. And when you add counter stain saffron in, these bacteria are stained pink in color. So when you see a slide which contains violet colored bacteria, it is gram positive bacteria, and when they are pink in color, they are gram negative bacteria. So this technique is widely used to classify bacteria. Now this is the final slide. Thank you very much for your patient listening, and please like and subscribe if you find this information useful. Please press the bell icon to receive more notifications, and thank you very much for your overwhelming response to my first presentation. Next week, I will come with another presentation for all of you. Thank you very much.